Hello viewer, welcome to my next lecture, and this lecture is related to pharmacodynamic methods. So if you remember, we have already talked about the bioavailability and its and the term of pharmacokinetics. So in this lecture, we will discuss about the pharmacokinetic methods, that is acute pharmacological response. So how we can get the acute pharmacological response. So the PK methods, they are inaccurate or non-reproducibles. Acute pharmacological effect like ECG or EEG readings, people diameter, etc., is related to the time course of a given drug. So I mean that the pharmacokinetic methods as compared to the pharmacodynamic methods and acute pharmacological responses, they are inaccurate and non-reproducibles. Acute pharmacological effect like ECG and EEG readings, people diameter. So the bioavailability is also determined by the construction of the pharmacological effect time curve as well as dose dependent graphs. The methods requires a measurement of response for at least 3 biological half-lives. There are some disadvantages, demerits related to pharmacodynamic methods. The pharmacological responses tends to be more variable, and accurate correlation between measured response and drug concentration is difficult. So I mean that in pharmacological response, you will have to see the variations and accurate correlations between the measured response and the drug concentration is difficult, and the observed response may be due to the active metabolite whose concentration is not considered for the pharmacological effects. And therapeutic response method is based on observing the clinical response to a drug formulations given to patients suffering from disease for which it is intended to be used. So in therapeutic response methods, there are some limitations in which there is improper quantification of observed response, and we should assume that physiological status of the patients doesn't change significantly over the duration of study. So a patient who required the drug for disease would be able to receive only single dose of a drug every week or a few days. Drug-drug interactions, it should be also under investigations. In vitro in vivo correlations. In vitro dissolutions alone will be insufficient to predict its therapeutic efficacy. So I mean that in correlations between the in vivo dissolution and in vitro bioavailability, so it must be established to predict its therapeutic efficacy. So I mean, if you want to determine the therapeutic efficacy of a drug, so we will have to correlate the in vitro dissolution study and in vivo bioavailability. It must be established in order to predict its therapeutic efficacy. In vivo and in vitro, it is a mathematical model that describes the relationship between an in vitro property, rate and extent of dissolution of the dosage form, and an in vivo response, the plasma drug concentration or the amount of drug absorbs. The objective of the developing IVIVC to enable the dissolution test to serve as alternates for in vivo bioavailability studies. Now there are some applications. You can see that there are certain advantages to ensure batch to batch consistency and therapeutic efficacy of a drug products based in vitro test. To develop a new dosage form with desired in vivo performance, validating the dissolution specification and development of BioWeber guidelines. You should also estimate the magnitude of the error in predicting the in vivo bioavailability results from in vitro dissolution data. Approaches which are used to establishing a linear relationship between the in vivo and in vitro parameter by using the data from the previous in vivo studies to modify the in vitro to develop the in vitro and in vivo dissolutions and in view bioavailability study. And correlation, it is claimed that it is based on the urinary excretion data. So, the dissolution parameters are related to the urinary drug parameters. So, based on pharmacological response, acute pharmacological effects such as LD50 is related to any of the dissolution parameters. And the statistical moments theory, it relate to MDT to MRT. Based on the plasma level data. So, here the linear relationship between the dissolution parameter and plasma level data are established. In vitro dissolution parameters, the time for the specific amount of drug to dissolve, for example, 50% of the dose, while in vivo plasma data parameter, the area under curve C maximum. So, here in vitro dissolution parameter, the amount dissolved at a specific time point, while in vitro dissolution, while in vivo plasma data parameter, the friction absorbed or absorption rate constant, equilibrium constant. In vitro dissolution parameter, mean dissolution time, and in vivo plasma data parameter, mean residence time, mean absorption time should be under investigations. The levels of correlations: level A, highest category correlation representing point to point relationship between the in vivo and in vitro parameters, while in vitro dissolution and in vivo absorption rates curve are superimposable. So, in this regards, the in vitro dissolution, the curve serve is alternate for in vivo testing and accurate predict its therapeutic efficacy. And level B, it is not a point to point correlation utilizing the principle of the statistical moment analysis. Here, mean dissolution time is compared to either the mean residence times or in vivo dissolution time. So, it cannot justify the changes in manufacturing or modification in the formula based on level B correlations, while in vitro data it cannot be used for the in vivo quality control standards. And level C, it is a single point correlation that relates on one dissolution time, point T50 to one pK parameters such as area under curve T maximum and C maximum, useful is guide in formulation development or QC. Multiple level C correlation involving one or several pK parameter to the amount of drug dissolved at various time point. Now, you can see here I am going to classify the drugs on the BCS basis. So, there is, you can see here on my screen, on my computer screen, that here the classification of drugs on BCS basis they are given. So, this is drug, for example, a drug having high permeability and a drug having high permeability and high absorption number. So, it come under the category of class 1 or class 2. Drug having low permeability and low absorption come under the category of class 2 and class 3. Further, the high permeability and high absorption number class 1 and class 2. So, the high absorption number class 1 and class 2, they are further divided into two. So, the class 1 has high dissolutions number or high dissolution profile, while the class 2 has low dissolution number, while in case of low permeability and low absorption number in class 3 and class 4. So, it is divided into two, and class 3, so class 3 has high dissolution number, while in case of class 4 it has low dissolution number. Look at the biopharmaceutics classification system for drugs. This is class 1, class 2, class 3, class 4. So, in class 1 you can see the solubility, and this is permeability, and this is the absorption pattern, and these are the example. So, in the first class and class first. So, and the class first solubility, the solubility and permeability is high, and it has high absorption pattern, will absorb pattern, and its example is diltiazem, propranolol and metoprolol. So challenges in drug delivery as the class 1, the CR form. The CR forms need to limit drug release or dissolution since the absorption of released drug is rapid. While in class 2, so a class 2 drug having low solubility and high permeability and have variable absorption patterns. So the classical examples are nifedipine and naproxen. So the challenges in drug delivery is to overcome the solubility and dissolution problem. While in class 3, so class 3 drugs have high solubility and low permeability having variable absorption patterns. The example is insulin and metformin anti-diabetic agents. So what are the challenges which is faced by the scientist and drug delivery? That is, so we should increase the permeability of these drugs. While in class 4 drug, it has low solubility and low permeability and poor absorption pattern is poor. So the example is Taxol and furosemides. So the challenges in drug delivery is we should improve both the dissolution and permeability. This is the biopharmaceutics classification system for the drugs. Now you can see here on the screen that BCS drugs for immediate release drug products in vitro and in vivo expectation. The class 1, that is called class 1, class 2, class 3 and class 4. So in class 1 it has high solubility and high permeability if the dissolution rate is slower than the gastric emptying rate. Then predicting the in vitro dissolution and in vivo bioavailability from dissolution data, that is okay, that is yes. So in class 2 drug having low solubility and high permeability. Low solubility and high permeability and in vitro dissolution rate is equal to the in vivo dissolution rate. So it is predicting that IV and IVC from dissolution data. Class 3 drug, according to the classification of biopharmaceutics, so class 3 drugs have high solubility and low permeability. So it means that the absorption is rate determining no IV and IVC from the dissolution data. So it means that in case of class 3, the drugs having high solubility and low permeability. So it means that its absorption rate is determining with no IV and IVC from the dissolution data. While in case of class 4, which having low solubility and low permeability, it is limited. So it means that in these two cases you can see here in class 1, BCS class 1 and class 2 drug, while in BCS, if we compare the BCS class 1 and class 1, class 2 and 3 and 4. So the BCS class 1 and class 2 having high solubility and high permeability. So it means, so that is predicting to the predicting to the dissolutions rate and bioavailability. While the BCS class 3 and class 4, so which have high solubility and low permeability. So in this case, this report is, you can see here, that is not properly predicting from the IV and IVC from the dissolution data. Inshallah, in my next lecture, we will talk about the bioequivalent study. So what is bioequivalent study? So we will discuss all the protocols, all the applications, merits and demerits of bioequivalent, why the bioequivalent study is important. Thank you so much for watching my lecture.